I'm Isaac. I'm a PhD candidate at the University of Melbourne. I'm here representing the Scanpy project, a toolkit for single-cell analysis. I'm going to talk a bit about the data that I work with, about the project itself, and then how this relates to Dask. So single-cell data is any data that's collected on individual cells. One of the more popular kinds of single-cell data is single-cell RNA sequencing data, or scRNA-seq. This allows us to look at which genes are being expressed in each cell. Each single-cell RNA-seq dataset will count the presence of tens of thousands of genes in thousands to millions of cells. This is represented as a big sparse matrix, which we can then analyze to find out things like what types of cells are there, what states are they in, and can we find relationships between these states. The top plot here is a histogram of the number of studies with single-cell data per month over the past few years. As the method has matured and gotten cheaper, it's been growing massively in popularity. The bottom plot is showing the relative sizes of these datasets. Early studies would only sequence tens or hundreds of cells, while contemporary datasets are characterizing thousands and millions. Not only that, but a hot topic of research is forming integrated atlases by combining many individual studies. All this is to say, the data is fairly big and it's only getting bigger. So that brings us to my project, Scanpy, which is a general toolkit for working with single-cell data, particularly single-cell RNA-seq. We provide functionality for many parts of the analysis. This includes pre-processing, things like feature selection and normalization, dimensionality reduction, visualization, as well as cell type identification and characterization. There are a few other libraries that do this kind of analysis. Most of them are in R. Apart from being in Python, one of the key differentiators that we have is the amount of emphasis that we've put on keeping these analyses fast and memory efficient. Scanpy is now at the center of a growing ecosystem for single-cell analysis in Python. It has over 400,000 downloads, a healthy community of contributors. Additionally, there's over 100 dependent packages as measured by GitHub. A lot of tools in this ecosystem do things like address new kinds of modalities or have new ways of modeling the data. I think there are two things that are making this ecosystem successful. First is heavy use of the PyData ecosystem. You have all this great stuff to build on, and a lot of our users are already familiar with these APIs. The second is that we define this core data structure that represents the whole data set that makes moving it around between different tools quite easy. This is what I spend a lot of my time thinking about. This core data structure is called AnnData, short for annotated data, and at its core, it's an annotated matrix. You have your main data matrix X, whose core dimensions, the rows and columns, correspond to observations and variables, respectively. You can start adding annotations on top of these dimensions. You can start adding annotations and learned representations on top of these dimensions. This is essentially a collection of dense arrays, sparse arrays, and data frames aligned to core dimensions. We also defined an on-disk schema, so you can take this data set and write it to HDF5 and ZAR stores. So in practice, what does this end up looking like in practice? So you have your AnnData object, which has an in-memory representation. It can be passed around and between a bunch of other tools, which makes the analyses using it quite composable. This dataset can then be moved transparently and quickly between disk and memory, where the on-disk representation can be used by a number of tools outside of the Python ecosystem. So some examples of these tools include CellByGene and Viteshi, which are web-based viewers, which are super useful for letting your less technical collaborators interrogate the data. There's also projects in the R ecosystem, which are able to read and write AnnData files. You can upload these files to web servers like Galaxy, where you can then do a lot of analysis through their online tools. And datasets are also distributed as AnnData files. So for example, CZI and the Sanger Institute's COVID-19 cell atlas made a bunch of datasets viewable via CellByGene and downloadable as AnnData files. So we'd like to integrate Dask with Scanpy and AnnData. Why would we want to do this? Well, datasets are big and they're only getting bigger. We also build on a lot of the same technologies, which should hopefully make integration pretty easy. Dask also represents a full solution for scaling out, where we don't have to roll our own out-of-core computation, which is something that we have done previously. It's also got a pretty powerful ecosystem, so hopefully we can really latch on to these already made cool tools. So why aren't we integrated with Dask already? Well, we have some challenges and some questions. Our biggest challenge is probably sparse data. Our data is really sparse. While there may be around 20,000 variables in the typical data set, we only see a couple thousand of these per cell. So CSC and CSR sparse matrices are pretty core to our package, and we're going to need good support for them before we can have tight integration with Dask. This is something that I'm trying to implement upstream and would love help with, and would also love to talk to people about. We also have an existing on-disk format. It's a nice, accessible schema, I think, but we're unsure how easy it'll be to make it play well with Dask. For example, we have data frames which are represented as like HDF5 groups, and then in a columnar format. Can we make Dask load these efficiently can Dask load these efficiently without having to do a bunch of extra computation? We'd also like to learn about other people's experiences, especially people who have been integrating Dask with pre-existing libraries and hearing about how well that went. Thanks for watching this video, and I'm really looking forward to meeting people at the summit.